Nandita Patadia, Assistant Professor in Microbiology Department at LJ Institute of Applied Sciences, LJ Campus, Ahmedabad. And today we are going to discuss a practical session regarding blood grouping, determination of human blood group by ABO and RH system. Discovered by Carl Linz Sr. He grouped the blood group of human into 4 groups based on the antigen present on the surfaces of the RBC, that is red blood cell. This was known as the ABO system. Later on, other blood group systems also developed like RH, MNS, P, Lutheran, Lewis, Kidd, Duffy, etc. But ABO and RH blood groups are considered as a major blood group which are important during the blood transfusion as they create complication during the transfusion of the blood. Other blood groups are also considered, but it is a minor blood groups. The blood incompatibilities are due to the reaction between the antigen present on the RBC and the antibody present in the recipient's blood, which will lead to hemolysis. See, blood group A is considered as the antigen present on the RBC is A and H antigen, and that person is having antibody B. So he can donate blood to, or he or she can donate blood to A and AB both. While the person having antigen B and H on the RBC having antibodies against A. So antibody A, antigen against antibody against A antigen. In case of O blood group, antigen is not present. Any antigen is not present. Only H antigen is present. A and B antigen is not present. In that case, the human's blood is having antibody against B and A antigen. Next is the Bombay phenotype. That type of RBCs are not having even H antigen. So the person is having antibody against H, A and B. All the types of antibody, and he is not having any antigen. See, here you can see that group A, B and AB and O. AB blood group is having both the types of antigens, so his blood is not having any type of antibodies. Another system is RH blood group system. The RH blood groups are secondary significant during blood transfusion. It is based on the presence and absence of RH antigen on antibody and RBC, and designed as a RH positive or designated as RH positive and RH negative. The antigen was first discovered or detected on the RBC of rhesus monkey, and hence it is the name has been given as RH antigen. When RH negative individual receive the blood from the RH positive donor, it stimulates the production of anti-RH antibodies of IgG class, and these antibodies are responsible for blood group incompatibilities. So this blood group antigen is also responsible for severe hemolytic disorder in newborn babies and erythroblastoma fetalis when the mother is RH negative. So mother is having the antibody against RH antigen and child is RH positive during the second or subsequent pregnancy. So what will happen? The mother's antibody will pass through the placenta to the child or fetus, and hence it chance to development of RH anti-RH antibodies are responsible for the disease of like erythroblastoma fetalis. Third is the Bombay phenotype, that is important. First reported by Bende and et al. In Bombay in 1952. The H antigen is foundation upon which A and B antigens are built. So absence of H, A and B antigen, no agglutination with anti-A, anti-B and anti-H antigen antibodies means no antigen is present, not a single antigen is present. That is called Bombay phenotype, and incompatible with any ABO blood group and compatible with Bombay phenotype only. So Bombay phenotype blood group can donate and can accept from the same only. They are not compatible with ABO blood groups. What is the principle of this technique? Human red blood cells possessing A, B and D antigen will agglutinate in presence of antibody directed towards the respective antigen. And agglutination of red blood cells is a positive test result and indicate presence of corresponding antigen. For that we require 0.5 ml normal saline, pricking needle or lancet, alcohol for the fingers tip sterilization or surface sterilization of finger, cavity slide, antisera A, B and D. Here in picture you can see that antisera bottles are available in market like this, and microscope for macroscopic observation of agglutination. You can observe the agglutination reaction macroscopically by naked eye, and if you want to further observe under microscope, you can observe under microscope also. Procedure: how will you perform this blood grouping? Take a clean glass cavity slide and mark with the A, B and D. Disinfect the fingertip with the 70% alcohol. Allow the alcohol to air dry and then prick with the pricking needle and make a puncture and collect the capillary blood. Before collecting, please wipe first drop of the blood, and then after that, by slightly squeezing, develop the second blood drop for the collection. Now collect 2 to 3 blood drops in the test tube, or you can directly collect on the cavity slide also. When you are collecting 2 to 3 blood drops in the test tube, you have to add normal saline and mix it well. Now from the test tube, add 1 drop in each labeled cavity A, B and D. And if you are collecting blood drop directly in the cavity slide, then you have to add single drop in each blood drop with the normal saline. Mix it thoroughly. Now add 1 drop of antisera A, B and D respectively labeled cavities having blood drop with a normal saline, and allow it to react and mix it gently and thoroughly. Rotate it slightly for 1 to 2 minutes, and after that observe the clumping in the circle. Interpretation of the result is done like this. If the anti-A is showing positive result and B is showing negative and D is showing negative, then the blood group is A negative. Why? Because RH is giving negative result. If A circle is giving positive result, B cavity is giving negative result, and D cavity is positive, then blood group is A positive. Respectively, positive and negative we can identify. Here in this picture you can see that this is the picture of blood group A, blood group B, blood group A, B both positive, A and B, and here only D. So this is the O positive, A negative, B negative, only D is showing clumping. Here only A is showing clumping. Here only B is showing clumping. Here A and B both the showing clumping, and here only D is showing clumping. So this is the A, blood group A, blood group B, blood group AB and blood group O. This picture is showing that neither A nor B and RH is also not showing clumping. So the blood group is O negative. So in this way you have to observe the results. So I hope all of you are now clear with the discussion. Let's begin the performance of the detection of human blood group by ABO and RS system. For that, you require antisera. This is readily available in the market. You can see that antisera A, B and center is having B colorless, A is of blue color, B is having yellow color and D is having colorless. We are having this normal saline over here, and this is the pricking needle, and this is the alcohol swabs. Now we are having 3 cavity slide. We have already marked it as A, B and D. So A, B and D mark slide we are having. Before using, just clean the slides and cavity. You have to just clean your finger tip like this. Allow it to dry. Now open the needle. Once again we are cleaning it. Just 30 seconds you have to wait for drying. Now prick the fingertip like this. So first drop will come like you have to wipe the first blood drop. Allow blood to form a drop and then collect in the cavity slide. We have to collect 3 drops of blood in respective labeled circle. Now we are adding 1 drop of normal saline in each drop. Now mix it the blood as well as normal saline. You can use this applicator or mixing stick. After adding antisera, you have to use different applicator stick. Now we are adding antisera. See, this type of blood drop you have to collect. Now, this is antisera A. When you are adding antisera, be careful, add it with the distance, don't allow to touch with the blood drop. Only 1 drop of antisera is enough. Antisera B. And finally antisera D. You can rotate with the slide with the drop, mix it with the rotating, or you can use this applicator stick for the mixing purpose, but at this time you have to use different stick for each cavity. Now mix it well and rotate it. See, results are there. You cannot observe or the clumping is not there with the antisera A. Here we can see that clumps are there in the B. And this is antisera D is also showing clumps. See, you can now visualize the clump. See, B is showing the clumps, D is also showing the clumps, and A is not having any clumps. You can see that fluffy clumps are there. B. So we can say that the test sample is having blood group B positive.